So without further ado, it is my great pleasure and honor to introduce to you Dr. Andre Vinicius Florentino. Yay, Dr. Florentino. Thank you from our Brazilian crowd there too. Dr. Andre Vinicius Florentino is a Brazilian gynecologist, professor, author, and entrepreneur. He holds a master's degree in health sciences with postgraduate training in nutrition, gynecology, longevity, health leadership. Dr. Florentino served as assistant professor of gynecology at the Federal University of Campina Grande in Brazil. Dr. Florentino is vice president of the Brazilian Society of Personalized Medicine. He is recognized for his work in precision-based hormone therapies and integrative approaches to women's health in midlife, integrated gynecology, metabolism, gut microbiome science, and lifestyle medicine. He is the founder of several medical education initiatives in menopause and endometriosis. Welcome, Dr. Florentino.
Thank you very much. Good morning, everyone. Good morning. Thank you, Dr. Mahmoud Kareishi, for this kind introduction. It's truly a pleasure and the honor of a lifetime to be here with you all. I would like to sincerely thank the organizing committee, especially you, Dr. Mahmoud Kareishi, Dr. Deborah Kolick, Dr. Tyler Ear, Sarah V, and the entire WEM team for putting together this amazing conference with so many outstanding faculties. It's also a great pleasure for me to see so many familiar faces from my Brazilian mentees from Hormones Mentoring. Thank you all for being here. And it's going to be a nice day today. I want to discuss the topic that, in my view, represents one of the most exciting emerging frontiers in women's health, the interaction between the gut microbiome and menopause physiology. For decades, our clinical discussions around menopause have focused primarily on hormones. We talked about estrogen levels, progesterone therapy, doses, routes of administration, and treatment protocols. But we are now realizing that another powerful biological system is also deeply involved in how women experience hormonal transition, and that's where the gut microbiome plays a great role. So raise your hand if you are familiar to this topic. Not you, my Brazilian friends who have already watched this presentation so many times. That's great. So here are my disclosures, and none of them will have to do with my talk today. And before we begin, here are the learning objectives upon completion. I want you all to understand the gut microbiome changes in menopause, the hormonal impact caused by the estrobolome, the relationship between an abnormal estrobolome and estrogen-dependent diseases, to understand beta-glucuronidase and also glucuronidation, and of course, how to support the microbiome. And I want to start this talk by highlighting that the role of the gut microbiome, especially in menopausal women, has gradually attracted researchers' attention. So how rapidly has this field grown? To answer this question more clearly, we have to examine the publication's trends. And if we look at the trends in PubMed, something very interesting appears. Before around 2016, as you can see here, the number of studies linking the gut microbiome to menopause physiology was extremely low. However, after 2020, we see a dramatic increase in publications, and especially in the last few years. This surge reflects that we are entering a new phase of research in menopause, since the microbial metabolism may become just as relevant as the endocrine physiology. So for those who are newer to this topic, I just want to give you some background information so we are all on the same page. Let me briefly review some essential definitions. So microbiota refers to the collection of all microorganisms residing in and on the body, including bacteria, fungi, viruses, whereas microbiome stands for the aggregate of all the genetic material of the certain microbiota. Whereas estrobolome is the collection of genes in the gut microbiota responsible for estrogen metabolism. Metabolome is the total collection of metabolites in a given environment, and dysbiosis, and we're going to talk about a lot of this, corresponds to the imbalance or impairment of the microbiota, and it's characterized by either the gain of pathogenic microbes or the loss of probiotics. But among all of these definitions, one concept is particularly important for our discussion today, and we're going to focus our attention on the estrobolome, which corresponds to phase 3 estrogen metabolism. Hold this information for now. So to better understand the interaction, we must also examine the factors that can shape the gut microbiome itself. So the gut microbiome is not something static. It is shaped by multiple environmental and physiological factors that can include genetics, infection, stress, alcohol intake, exposure to chemotherapy, radiation, the use of antibiotics, the pattern of diet, even your geographical location, the use of pre- and probiotics, and hygiene. And these regulatory influences can lead to a downstream physiological consequence throughout your entire body. Once established, the microbiome exerts, as you can see over here, a broad systemic effect. Microbial metabolites can influence signaling, metabolic regulation, endocrine pathways, and even the production of neurotransmitters. This helps to explain why dysbiosis has been linked to so many diseases like metabolic disease, autoimmune disease, and even neuropsychiatric conditions. So, as you can see, the gut microbes, they're critical for maintaining host health. This figure, I just love this figure because it illustrates a fundamental concept. The gut microbiome is not something we simply acquire and that keeps stable throughout life. It's a dynamic ecosystem that begins to develop at birth during the gestation and continues to evolve through your entire life. The first major determinant of microbial composition occurs during delivery. So as an OBGYN, we have to be conscious because a C-section and a vaginal delivery triggers different profiles of colonization. During infancy, feeding patterns can play a central role. If the baby is breastfed or bottle-fed, it will change the microbiome. And by the time a child reaches approximately 2 to 3 years of age, the gut microbiome begins to resemble the adult configuration. However, its composition continues to be influenced by factors such as diet quality, antibiotic exposure, host genetics, and even environmental interactions. So one way to appreciate the biological significance of the microbiome is by looking at its genetic capacity. It is estimated that microbial cells in human body exist in approximately 1-to-1 ratio with human cells. Although the number of microbial genes surpasses human genes by a ratio greater than 100-to-1, reflecting this vast genetic diversity of the microbiome. Just for you to know, we have around 23,000 genes in our human genome, in contrast to 22 million genes in our gut microbiome. The human microbiome has revealed extensive taxonomic and functional diversity, as well as significant variations across body sites and between individuals. Another important concept for you to keep in mind is that the microbiome is not uniform throughout the body. Different anatomical sites can harbor distinct microbial ecosystem, each one adapted to its local environment and also to their local function. For example, as you can see in your right-hand side, there is a predominance of Firmicutes in the vagina in contrast to a predominance of actinobacteria in the hair. One interesting observation in microbiome research is the shift that microbial composition occurs after menopause. Studies have shown that postmenopausal women tend to exhibit a Firmicutes to Bacteroidetes ratio which begins to resemble the microbiome pattern seen in men. And this is important because these microbial shifts have been associated to metabolic changes including increased adiposity, the menopause belly, and altered glucose metabolism. So menopause is not only an endocrine transition, it's also a microbiome transition as well, as you can see here. During menopause, the diversity of the gut microbiome and the functional potential of the estrobolome, they are both reduced. Additionally, decline in estradiol and progesterone may compromise the intestinal barrier integrity, allowing microbial translocation into the bloodstream. And of course, all of us are well-trained physicians, and most of us were taught to think about estradiol and progesterone primarily related to reproductive organs. But both of them also maintain and improve the gut barrier function. And this interaction becomes particularly important when we examine the intestinal barrier. The intestinal barrier is notably thin. It is essentially composed of a single layer of epithelial cells. But despite its thinness, it serves as one of the most important interfaces between the external environment and the internal immune system. Every single day, this barrier is exposed to dietary antigens, microbial metabolites, and environmental toxins. And maintaining its integrity is therefore essential to prevent excessive immune activation and systemic inflammation. In a homeostasis state, as you can see here, is when you have the balance between the function of the cells in the epithelial lining of the gut and the immune responses to microbes. You see this thick mucus layer, an abundance of short-chain fatty acids, a microbial diversity, and a beneficial microbiota predominance. But you must be wondering, what's going to happen if this barrier becomes compromised? And that's dysbiosis. In a dysbiotic environment, microbial composition shifts towards what may produce these inflammatory metabolites. So this change can weaken the epithelial tight junction and also increase the intestinal permeability. When this occurs, microbial products such as lipopolysaccharides can enter circulation and trigger systemic immune responses. This phenomenon has been referred as the metabolic endotoxemia. What you can see here with this shift is that mucus layer is lost, and epithelial cells tight junctions, they are damaged and they become leaky to harmful microbiota. I don't know if you are familiar with the tight junction, but it's a structure responsible for controlling antigen trafficking and inflammatory response. Tight junction act as a molecular gatekeeper between intestinal epithelial cells. They regulate the passage of molecules between the intestinal lumen and the bloodstream. When tight junction integrity is preserved, the intestinal barrier becomes effectively. But when these structures are disrupted, intestinal permeability increases and inflammatory signaling may be amplified. So let me just show you with a very interesting video over here.
One of the most essential structures for barrier function are the tight junctions near the apex of the epithelial cells, which regulate the flow of fluid and salts. Each tight junction is a dynamic complex of more than 50 proteins that spans the epithelial cell walls. Outside the cell, the exposed loops of claudin and occludin proteins interact with loops of adjacent cells. Inside the cell, zonulin family proteins anchor the complex to the belt-like apical rings of the cytoskeleton. Contractions of these actomyosin rings tighten the gaps between cells and seal out damaging substances, including microbial components from the intestinal lumen, and keep the intestine's immune system in check. Cell signaling controls how the tight junctions form and remodel in response to different stimuli, part of the gut's natural process to maintain the barrier. Studies have shown that bacterial toxins and other stress.
It's amazing, isn't it?
Liam.
So, and regarding inflammation, there is this editorial publication in Menopause questioning. Inflammaging or inflammopause? Is inflammation the cause or consequence of vasomotor symptoms? So beginning as early as the 4th decade in both men and women, an aging-related chronic low-grade inflammation is known to start. Inflammaging, and I quote Mark, is characterized by rising pro-inflammatory cytokines attributed to accelerating immune senescence. At the same time, women may be especially burdened since the menopause transition, and the associated decline in estrogen can contribute to amplification of this inflammatory process. This amazing figure over here, and take a picture of it because it's amazing. During menopausal transition, declining estradiol levels, they are associated with increased production of pro-inflammatory cytokines, as you can see over there, interleukin-1 beta, interleukin-2, TNF-alpha, and these cytokines can cross the blood-brain barrier and activate microglial cells, triggering inflammation. That's what they call inflammopause. Right, eh? And this neuroinflammatory process may contribute to symptoms such as cognitive decline, mood disturbances, sleep disruption, and thermoregulatory instability, which corresponds to the main and the most common symptoms of our menopausal patients. This bidirectional communication between the gut microbiome and the brain is mediated by the immune system, the vagus nerve, the enteric nervous system, and the neuroendocrine system. Alterations in the gut microbiota have also been linked to conditions such as autism spectrum disorder, anxiety, depressive-like behavior, and impairment even in physical performance and motivation. This particular diagram is from a publication in Maturitas, and it illustrates how gut dysbiosis and the estrobolome dysfunction may contribute to several estrogen-related diseases, highlighting that the gut dysbiosis has positive correlation with the development of diseases such as endometriosis, PCOS, endometrial hyperplasia, obesity, metabolic syndrome, endometrial cancer, breast cancer, gastric cancer, and brain function. So here you can see the references below in this table. If you put the original article, you can get the expanded version of the reference for everyone. The gastrointestinal microbiota plays a potentially significant but yet enigmatic role in human health through the estrobolome. It is inevitable to say that Plotter and Blazer defined the estrobolome as the aggregate of bacterial genes whose products are capable of metabolizing hormones. Remember that I told you about this? So we have to remember that estrobolome is phase 3 estrogen metabolism. So let me walk you through the estrogen metabolism. Estrogens produced either by ovaries, adrenal glands, or adipose tissue. They are transported to the liver to be metabolized into a biologically inactive conjugated form. And this biotransformation occurs through 2 major phases. During phase 1, as you can see on your left-hand side, reactions generate intermediary metabolites through oxidative pathways. During phase 2 reactions, they are conjugating these metabolites, making them more water-soluble so they can be excreted either through feces, bile, or stool. In the bottom, you can see all the nutrients required for these reactions to occur. So among many metabolic reactions involved, especially in phase 2 estrogen metabolism, one specific deserves our attention, and that's glucuronidation. During phase 2 estrogen metabolism, a specific enzyme called UGT, they attach a glucuronic acid to a variety of toxins and hormones, typically inactivating and marking them for excretion. So glucuronidation is used by the body to eliminate hormones such as estrogen, testosterone, progesterone, xenobiotics as pesticides and herbicides, and our daily medication like oral contraceptive pills, Tylenol, even benzodiazepines. So here's the analogy for you to understand this reaction. Picture glucuronidation as this box. So the liver get things he wants to eliminate all packaged up. It puts the lid on top, ties this nice bow, and sends it to the gut. However, once these conjugated molecules reach the intestine, they encounter bacterial enzymes capable of reversing this process. So in the gut, beta-glucuronidase can untie the bowl, opens the lid, and release all toxins and hormones back to circulation. And this is where the gut microbiome becomes a crucial regulator of systemic estrogen level. So here you can see the entire picture of the estrogen metabolism, but I want to zoom it for you, because this raises an important question. When we consider the role of beta-glucuronidase, the key issue is not simply where the enzyme exists. The question is how active it is. Because when beta-glucuronidase activity is high, it will remove the glucuronic acid group that the liver just attached during detoxification. And by doing so, they regenerate the active form of estrogen molecule. And this allows estrogen to be reabsorbed into circulation, increasing estrogen total load within the body. On the other hand, when we have this active low, the estrogen will remain conjugated and will be released through feces, decreasing estrogen concentrations. So here are the main reasons why beta-glucuronidase activity can be high. The top reason is bacterial overgrowth, dysbiosis, but also liver stress, as Dr. Melinda Ring just addressed. The antibiotic use, dietary factors such as this Western diet enriched with high protein, high fat, low fiber, and the lack of beneficial flora, because we know that when you consume fiber and produce short-chain fatty acids, you acidify the colon. And lower colonic pH can decrease beta-glucuronidase activity. In contrast, when you have this higher colonic pH, that you increase beta-glucuronidase activity. Here you can see the major subset of beta-glucuronidase bacteria producers. So Escherichia coli, Ruminococcus, Bacteroidetes, Eubacterium, Peptostreptococcus, Staphylococcus, and Clostridium perfringens. These are the major subsets. And remember this, because we're going to talk about this a little bit further when we talk about stool testing. But you might be asking yourself so far, so Dr. Andre, is it good or bad to have beta-glucuronidase activity, isn't it? So the answer to that is you must remember the Paracelsus principle. The difference between medicine and poison is the dose. So in a healthy condition, your gut microbiota will work to promote balance. So in your left-hand side in the upper, if the woman has a lower estrogen level and she's in homeostasis, this will change the microbiota composition to increase beta-glucuronidase activity. So she will deconjugate more and reabsorb more estrogen in order to increase her own estrogen levels. Conversely, when a woman has a higher estrogen level, the gut microbial composition will decrease beta-glucuronidase activity, so it will remain this estrogen conjugated, releasing through feces. In contrast to all of that, when you have dysbiosis, there's no balance. In fact, women with low levels will remain feeling what you can see here in your right-hand side, the estrogen deprivation symptoms like insomnia, depression, anxiety, hot flashes, and also increasing risk factors, as you can see in your left-hand side, for abnormal conditions such as breast cancer, ovarian cancer, endometrial cancer, and even endometriosis. Now, let me just bring you some data regarding this topic. I would like to bring in a series of scientific publications to illustrate what the current literature is saying about what we have discussed so far. So an enriched astrobolum with genes encoding enzymes that promote deconjugation can facilitate the reabsorption of free estrogen, contributing to the total load of the host. This other publication provides insights into the fact that disruption of the microbiome may, however, be associated with an increased risk of developing inflammatory, autoimmune, and even malignant diseases. The human gut microbiome is functional and exerts both local and long-distance effects involving hormonal intermediaries, metabolites, and immune messengers. This one, I really love this study. It shows how much estrogen is truly eliminated versus recirculated. So studies with radio-labeled estradiol and estrone injected into women have shown that although a large proportion of the injected estrogen is excreted in the bile, approximately 50%, a much smaller fraction, around 7 to 15%, is found in the conjugated form in feces, indicating that a significant proportion of estrogen is really reabsorbed into circulation via the conjugation activity of the gut microbiome. Estrogen levels have been also linked to the gut microbiota. And therefore, changes in the gut flora may be associated with menopausal changes and even with diseases such as osteoporosis, metabolic disorders, depression, Alzheimer's disease, and neurogenital symptoms. Recent studies have also suggested a connection between ovarian aging and the gut microbiota. And changes in the composition and functional profile of the gut microbiota have profound consequences of ovarian function. There is a specific strain called Ingertella lenta that causes fibrosis and can induce premature ovarian insufficiency. So there is research on that as well. Recent advancement underscores the critical role of gut microbial homeostasis in female health, with dysbiosis potentially leading to diseases among women such as endometriosis, we are going to talk about this later today, breast cancer, and cervical cancer. Research on the gut microbiome is driven by its strong translational potential to use the gut microbiome as a disease biomarker, as well as the possibility of modulating the intestinal microbiota to alter not only the onset, but also the progression of disease. The reduction of strain Rosiburia plays a role in both the new onset or progression of osteoporosis. And for you, Dr. Corinne Mann and Dr. Avram Blumen, regarding the tamoxifen metabolism, that's very interesting. There's a publication from Endocrinology 2022 that says that the gut microbiota plays a crucial role in tamoxifen metabolism, as it facilitates the conversion of tamoxifen into active metabolites for hydroxytamoxifen and endoxifen, enabling its anti-tumor activity. So an imbalance of the gut microbiota may lead to abnormal metabolism of tamoxifen, affecting even the generation of its active metabolites, and thereby, if you have dysbiosis, you can reduce the efficacy of this endocrine therapy. This is a slide I put for Dr. Rachel Rubin and so many of you who are an advocate for local estrogen therapy. It's a climateric publication that highlights the fact that vaginal estrogen can restore the vaginal epithelial integrity. It can increase the mucus layer, increase the lactobacillus, and changes the microbiota diversity into a more beneficial one. So growing evidence has confirmed this bidirectional relationship between the gut and hormones, in which the microbiota acts as a regulator. So not only sex hormones, they serve as a substrate for microbial metabolism, but also the microbiota can influence the sex hormone levels through deconjugation. And this recent study, Dr. Deborah Kolek just sent it me a few days ago, a couple of months ago. These are very recent studies that have begun expanding our understanding of microbial hormone metabolism. So researchers are now identifying bacterial pathways capable of metabolizing not only estrogens, but also steroid hormones like testosterone. This is a 2026 publication. Much like the estrobolome, the testobolome may also be influencing the total levels of androgen. And the progestabolome may also influence progesterone and progestine's metabolism. So it's impossible to practice medicine nowadays without taking into consideration the impact of the gut microbiota. And it's funny, since I'm here in the US, to say that unlike Vegas, what happens in the gut doesn't stay in the gut. Because we have several microbial axes described in the literature. Each of these axes represents a communication network through which microbial metabolism influences systemic physiological functions. These pathways help explain the growing number of diseases now associated with microbiome alteration. For example, PCOS, adenomyosis, endometriosis, ovarian cancer, IBD, IBS, reactive arthritis, rheumatoid arthritis, and so on. So gut beta-glucuronidase is emerging as a new therapeutic target for menopausal women. And this is another recent publication from Gut Microbes 2023. So here are my clinical pearls for correcting beta-glucuronidase. Up to this point, we have established that the microbiome is not confined to a single organ. And this raises an important question. How can we, as clinicians, begin to modulate this process in a practical and targeted way? So here are what I do in my daily practice. First of all, check the oral cavity. For the ones who are not aware of, the oral microbiome is the second largest microbial community in the human body, followed by the gut microbiome. So check for the oral cavity. We have to remember that it's the beginning of the GI tract, so search for tooth infections. Address any bacterial overgrowth or digestive complaints. We have to remember that using non-invasive hydrogen or methane breath tests, you can diagnose SIBO and treat it accordingly. Support the growth of short-chain producing bacteria, especially through dietary interventions. I do agree with what Dr. Melinda Ring just said. Address your patients to augment their citrus ingestion, cruciferous vegetables, because they can provide glucaric acid. After you treat these bioses, you can consider, and I'm going to talk more about this in our panel regarding supplements. If you are going to supplement probiotics, literature shows that you have to consider to prescribe greater than 20 billion CFUs with at least 6 different strains that has to include lactobacillus species and bifidobacterium species in order to correct this balance. And sometimes consider supplements that can inhibit beta-glucuronidase activity. This one I particularly use very often, and so a lot of my colleagues here. It's calcium deglucarate from doses ranges from 500 up to 3 grams a day, because it can block the beta-glucuronidase activity while you are searching for the reasons why, connecting the dots to find the root causes. And here is another very nice figure, because there are 8 rules for gut health. First, mindful eating. Pause and think. Remember that mastication is a vital function that ensures that indigested food is broken down into pieces and be prepared for digestion. Avoid to eat food in front of a TV while you're looking at your Instagram posts. So mindful, it's something that I really address you to do it. Diverse food, at least 3 plants a week. There is a very nice study from NIH saying that if you can eat at least 3 different plants a week, it's great for your microbiome. Fermented foods, if possible, at least 3 portions a day. Pivot your protein, diversify sources, and for sure, quality over calories. You should eat whole foods. Avoid ultra-processed foods and eat the rainbow. The more colorful your plate is, the more enriched with polyphenols it is. And every time that it's possible, fast. Fast for 14 up to 16 hours, or at least anticipate your last meal. And you also would ask your patient to take a look and see what's her Bristol stool type. Yesterday, Dr. Steller taught us about this. So this is a low-cost, real-time biomarker of the gut health. So what we are aiming is type 4. This is smooth, well-formed stool, which suggests an optimal gut function, adequate fiber intake, and a more balanced microbiome. What about stool testing? Stool testing can give us a functional window into this ecosystem, allowing us to quantify microbial composition and, more importantly, to identify key enzymatic players such as beta-glucuronidase, which directly influence estrogen recirculation. So, do you remind that when I showed the major subset of beta-glucuronidase producers? So here in your right-hand side, these are exactly the ones that are elevated in this microbiome test. So you can check instead of guess sometimes. And you also can see the same test in this particular patient, an elevation in beta-glucuronidase enzyme activity and also zonulin levels. And we have so many more gut tests available for you to buy. This is another one called the Zoom Gutter and the Gut Zoomer. And you have the same pattern, high beta-glucuronidase, high zonulin levels. But there is something very important for you to remember. When we perform a single stool test, we are essentially capturing a snapshot, not the full movie, because the microbiome is very dynamic, because it changes when you eat and depending on what you're eating, and also when you restrict your diet. So to be accurate, you need a frequent sampling to account for this dynamic nature of a changing microbial system. And a few days ago, I was just reading that there is an Australian company that is developing a device that you can put on your toilet that continuously collects stool samples, I know, and use artificial intelligence to analyze your gut microbiome, allowing this real-time assessment of microbiota changes. So you must be wondering, if the gut microbiome is this dynamic, then the real question becomes, can we actually influence it in a meaningful and clinical